Hello, I'm Jan Zrimets and I'm a postdoc in the group of Alexei Zelezniak. And I'll present our recent research about learning the regulatory grammar of DNA for protein expression engineering. So, understanding the regulatory code of gene expression, as well as being able to manipulate it, can help us fine-tune how cells function, and this is important for solving problems in medicine and biotechnology. In the process of expression of DNA to RNA to protein, it's been shown across multiple organisms that there's a strong agreement between protein abundance levels and mRNA levels. And this suggests that actually transcription is the main determinant of protein abundance. So, focusing on transcription, we obtained practically all available RNA sequencing experiments, over 3,000 at that time, which also covered the majority of conditions. And this data is plotted in the figure. So, if we sort the protein coding genes on the y-axis according to the increasing average level of gene expression, we see that the variation of expression per gene across the conditions is actually much smaller than the whole dynamic range of expression levels. And this shows that genes that are highly expressed in the majority of conditions will likely be highly expressed, and vice versa for low ones. But it also suggests that this conserved basal average level of gene expression is actually encoded in the DNA sequence itself. So to test this, we needed a modeling framework that's able to extract information from DNA sequence. And luckily, in the past years, there's been a lot of progress with deep neural networks, which can learn to decode information in very complex images and can then translate this to human understandable language. So we can use same networks also to analyze DNA sequence. For instance, convolutional networks can learn to recognize the occurrence of motifs and groups of motifs, whereas recurrent networks can learn the sequential information in a DNA. However, recurrent networks are pretty hard to train, and with certain tweaks, convolutional networks can achieve similar results. So in the end, we used just stacks of convolutional networks with certain dense classical layers on top. So to train our networks and test how well we could predict gene expression levels from DNA sequence, we use this whole gene regulatory structure. So not just the coding regions, which were described with codon frequencies, but also the upstream and downstream sequences, which covered promoter regions, untranslated regions, as well as terminators, and spanned over 2,000 base pairs of DNA sequence. We also used some additional variables related to mRNA stability. So the trained models with data from yeast could explain over 82% of the variation of gene expression levels. We then trained also additional models with data from other model organisms spanning from bacteria to human and covering practically the whole range of genomic complexity. And this verified that this phenomenon occurs across all these organisms, where on average 60% of the variation of gene expression is actually encoded in the DNA sequence itself. So we then compared our models, our deep models, with shallow ones, so classical machine learning, as well as explored how the different regions contribute to the predictions. And we found two important points, that the deep models actually, the improvement of deep models is that they can extract information from DNA sequence much better than shallow ones, and that all of these different regions jointly contribute to the prediction of the gene expression levels. So each region carries a certain amount of new orthogonal information besides the large amount of overlapping information among the regions. And so to analyze this a little bit further, we performed analysis of mutation rates across orthologous genes from 14 related yeast species. And we found strong evidence that actually promoters and terminators co-evolve with their coding regions. And so this shows that this gene regulatory structure is not only interacting, but is actually a co-evolving unit. So then we were wondering what type of regulatory grammar these models learned. And for this we used a very simple approach, so sliding window occlusions in the input data, and we measured the output perturbations. So with this, we obtained the relevance for all of these regions in the input sequence data. And from this, we could extract the DNA motifs. And in these extracted motifs, we found that a lot of the known and expected DNA regulatory grammar from yeast was present, and this included transcription factor binding sites, Kozak sequences, as well as other motifs, so covering all of these different regions. We additionally performed association rule analysis, which is actually used in market basket research. So we treated each gene as a basket of motifs, and we then analyzed the co-occurrence of motifs across the genes. And we actually uncovered an over 8-fold larger amount of motif co-occurrence across the regions than in any single region alone. So if we look at an example of 4 genes that carry a common motif in their promoters, so a transcription factor binding site marked blue here, they also carry motifs in their other regions, marked red, which repurpose this motif to define a wide range of different expression levels. So if we then ask how much of the whole available range of expression levels we can recover, either with motifs or motif co-occurrence rules, we see that with motifs we can recover 57%, and with the rules 84%, which shows that actually motif co-occurrence rules can define practically the whole range of gene expression levels. So then finally we asked whether these models of DNA regulatory grammar could be used to guide gene expression engineering. And so considering the way people usually approach these problems of fine-tuning gene expression is that they focus on a single region like the promoter and just swap it out with certain known weak or strong variants. And they don't focus on any of the other regions which we showed can have quite a large effect on gene expression. So we took the genes and kept their native promoters fixed and varied the terminator regions with all the different variants available in our data set. And we found that actually there's a large amount of regulatory freedom per each gene, over 2 orders of magnitude, around their expression levels, which can be defined just by swapping out the terminators. And by keeping the terminators fixed and then varying the promoters, we obtained very similar results. So we then tested this also experimentally, and for this we designed constructs with native promoters and variants of terminators, either strong or weak, so either predicted to either upregulate or downregulate the gene expression levels. And we found that indeed, on average, over 20% of the gene expression could be varied with these different terminator regions. And we also found combinations of regions with much larger changes of expression levels. So this shows that just by learning from the native genome, we can guide gene expression engineering without the requirement of any synthetic approaches. So an older version of this study is available on bioRxiv, and hopefully the newer version will soon be published. It was a collaborative study between different departments and also the University of Gothenburg, and most of these co-authors are cited also on the paper. But I would especially like to thank Alexei for giving me the opportunity to perform this very interesting research. And thank you for your attention.